Hello, everybody. I'm Judy Aronchik, and on behalf of the Executive Council of the Association of Senior and Emeritus Faculty, I would like to welcome you all back to our virtual Luncheon Speakers series. You know that our lunches are very terrific, but we're not able to serve them to you now. However, we are still serving up terrific speakers, and today is no exception. Before I introduce our speaker, I would like to tell you about two very exciting upcoming ASEF events. On Thursday, February 18, you will have the opportunity to hear Dr. P.J. Brennan, who is our Chief Medical Officer and Senior Vice President at UPHS, who will speak on the very timely topic of vaccine distribution. And on Thursday, March 25th, we will have Dr. Anita Allen, who is our charismatic former Vice Provost for Faculty Affairs and expert on privacy law, and she will speak about issues related to privacy and COVID-19. Today, we are very fortunate to have Dr. Amita Sehgal with us. Dr. Sehgal is the John Herr Musser Professor of Neuroscience, investigator at the Howard Hughes Medical Institute, director of the Chronobiology and Sleep Institute at the University of Pennsylvania, and is currently the president of the Society for Research on Biological Rhythms. Dr. Sehgal received her Ph.D. from the Weill Graduate School of Cornell University and conducted her postgraduate work at Rockefeller University. She has made fundamental contributions towards our understanding of the molecular mechanisms that generate endogenous circadian rhythms and also the genetic mechanisms and functions of sleep. Dr. Sehgal has served on many national and international committees involved with neuroscience, from the NIH to Germany to Beijing. She has also served on editorial boards of many neuroscience and biological rhythms journals, including the Journal of Neuroscience and the Journal of Biological Rhythms. Dr. Sehgal has received many prestigious awards and honors, including the Outstanding Scientific Achievement Award from the Sleep Research Society and the Javits Investigator Award from the NIH. Today, she will speak to us on keeping ourselves fit by maintaining daily rhythms of sleep and activity. And it is my pleasure to turn my computer over to Dr. Sehgal.
All right, thanks so much for that nice introduction, and thank you for having me here. I'm gonna, oh wait, it says host disabled, participant screen sharing. Am I a host or no? I guess I'm not. Okay, now I'm the co-host. I think I just got made the host. Okay.
So you can share.
Okay, here we go. Can you see my screen?
Yes.
Okay, great. So yes, thank you so much for having me here. I'm happy to, share the work that we're doing. Also, I should say, I welcome questions along the way. So if anybody has something they want to ask, feel free to jump right in. I'd be happy to have this be a discussion rather than a lecture. So yeah, what this talk is going to be about is, some of my own work, but more about just what is going on in the fields right now, the fields of circadian rhythms and sleep. And I'll provide a little bit of background to tell you how we got to where we are today in these fields. So my title says sleep and activity, and that of course is the best known example of a circadian rhythm. But the truth of the matter is that most physiological processes cycle with the 24-hour rhythm. So shown here on this 24-hour clock are some of the processes that in humans change with time of day. You'll see here, cardiovascular strength, muscle strength, blood pressure, body temperature, melatonin, cortisol, lots of things cycle. The truth of the matter is that it's hard to find a physiological process that does not on some level show circadian regulation. In other words, that does not on some level change with time of day. So why should we care that things are going up and down? And the reason we should care is because when these rhythms are disrupted, and that can be through intrinsic factors like genetic mutations, or it happens with aging, or it could be extrinsic factors. Shift work is a big one. Jet lag, which maybe these days we're not traveling, so we're not suffering from it, but otherwise it can take a toll. even eating at the wrong time of day. And what those do, if you are susceptible to such circadian rhythm disruptions, is you're more at risk for neurological psychiatric disorders, some of which are listed here, and also more at risk for metabolic and cardiovascular disease. So the big question is, how are these rhythms generated? Now, the first official report recognizing that there were circadian rhythms was from this French astronomer who noticed in mimosa plants in 1729 that the leaves were opening and closing, and not just in constant conditions where there were light-dark cycles, but also in constant conditions where there wasn't a cycling environment anymore. And so he raised the idea of these rhythms being generated endogenously. Now, prior to that, the Swedish naturalist Linnaeus had also observed rhythms. But the idea that they were controlled by internal circadian clocks was not accepted till much later. I think maybe even like the 1940s. And the reason was that even though people had seen these things persist in constant conditions, like in constant light or in constant dark, the thinking was that they must be driven by some environmental cycle. So maybe not light dark, maybe it's, magnetic field or something like that that's changing. But that, the idea was that it has something to do with the Earth's rotation and there's an environmental signal. One of the things that convinced people that these clocks are internal was the finding that the internal circadian period is not the same in all species. And it doesn't precisely match the Earth's cycle. So we, for instance, don't have internal clocks of exactly 24 hours. So we're actually a little bit longer. And then there are some species like birds and flies that are a little bit shorter than 24. And so this finding that circadian period varies from species to species and, in most cases is not exactly matched to the environment was what led to the acceptance of the idea that these clocks are internal. And the first evidence that clocks are genetic came from my favorite model organism, which is the fruit fly Drosophila melanogaster. Now, back in the day, different assays were used to study circadian rhythms in Drosophila. The most popular one now is where we basically monitor their activity using a system such as the one shown here. So you place a fly in a glass tube, like here, project an infrared beam in, which the fly cannot see. When it's active, it marches back and forth and deflects the beam, generating events picked up by a computer, and then you get these actograms or activity records. So the shaded areas are activity, and you can see that the fly has a robust rhythm where it's active at some times of day and not at others over a 24-hour period. Now, the first genetic mutants that affect circadian rhythms were first found in Drosophila. And this initial work was done, pioneering work was done by Ron Kanapka, a graduate student in Seymour Benzer's lab at Caltech. And this was done published in 1971. Now, back then, they didn't have computers. So what they did was to still be, monitor activity of the fly. And they used a Esterline Angus, it's like a chart recorder. And every movement of the fly results in a deflection of on the chart recorder. And that is shown here. So this literally, each single tick here is a movement. And these are the same as the actograms generated by the computer. They're plotted a little differently here. So each line is 24 hours. So here's your wild type animal that has a period of 24 hours. Here's a mutant that he got. So he got every type of mutant you might want out of the screen. So here's the short period mutant that has a period of 19 hours. So each day activity begins and ends earlier than the day before. These, I should mention, are all done in constant conditions. So there's no cycling environment. This is the internal clock at work. And then here is another mutant that has a long period rhythm. So activity each day is later than the day before. And then finally, here's a mutant that's arrhythmic. And all of these mutations ended up mapping to the same gene, and that was the period or the PER gene. So you can mutate PER to change rhythms in pretty much every way possible. So PER became this really important gene. It was this canonical, a real paradigm for a behavioral gene where otherwise the animals looked okay, but the rhythms were totally off. Now, it wasn't until almost 25 years later, this was published in 1994, when I was a postdoctoral fellow with Michael Young at Rockefeller, that we found a second mutation that affects circadian rhythms in flies. So here again is the wild type animal active at the same time, every day. And then here is the mutant that we got, which we call timeless. And that is arrhythmic, that's essentially active at random times. And putting all of this together led to the model for how you make a clock within an organism. And here is the model in its very simplest rendition. And that is that these two genes, period and timeless, or PER and TIM, make RNA at a specific time of day. And then that RNA gets made into protein at a specific time of day. And that protein goes back to turn these genes off. Now, we always knew that the clock had to be a cyclic process because it has to be self-sustaining. And so this is the cyclic process. This loop takes 24 hours to complete. And it takes place with a short period in the short period mutant, and it takes place with the long period in the long period mutant. And when you're missing either of these genes, then the animal is totally arrhythmic. So this then is, now this is, like I said, a very, very simplified version. We now know that there are many more components that are part of this, and there's much more regulation at every step, but this is the simple model. And then it turned out that this model was conserved across species, and most of the genes are even conserved in mammals, to the point where people then started finding that mutations in these genes are associated with human disorders. So this is work from the Louis Potacek and Ingwei Fu labs, where they were studying people who had what's called familial advanced phase sleep phase syndrome. So these are people who are 4 or 5 hours advanced with respect to the rest of society. They go to bed at like 7 p.m., they wake up at 3 a.m. And these researchers were studying families that had these disorders. And they found the first family where they identified the genetic basis turned out to have a mutation in the PER2 gene. So this is the human homologue of that gene that I just told you about that was discovered in flies, the period gene. And it was in a phosphorylation site on the PER protein that they found this one mutation that accounts for this phenotype. A few years later, they found the molecular lesion in a second family. And that family turned out to have a mutation in the kinase that does the phosphorylating of PER. This again was a kinase that was first found in flies as part of the circadian clock. And then now it turned out to be associated with a human disorder that causes advanced sleep phase. So these genes that were found in the fly led to an understanding of how you generate a clock. And then now it's turning out that mutations in those same genes are causing human circadian disorders. So because of the relevance of this work, the 2017 Nobel Prize in Physiology or Medicine was awarded to the researchers that had started these studies, these molecular studies in Drosophila. And one of them was my postdoc mentor, Michael Young. So what do we know about clocks at this point? So, once we started cloning clock genes, what was done was to go see where they are expressed. Now, the thinking had always been that the clock would be in the brain. And from the brain, it's, controlling behavior and maybe some other things. So one of the surprises when these clock genes were isolated was that they actually are in all body tissues. And so yes, definitely the major clock is in the suprachiasmatic nucleus in the hypothalamus of the brain. And this is the only clock that receives information about the light-dark cycle through the retinohypothalamic tract. But there's a clock in muscle where it's regulating fatty acid metabolism, clock in adipose tissue where it's involved in adipogenesis and in lipid turnover, clock in the liver that's involved in cholesterol and glucose metabolism. And these clocks are kept synchronized to the day-night cycle by the clock in the brain. So there are multiple mechanisms, including circulating factors, released from the brain that are synchronizing all these clocks to the day-night cycle. Now, how do these clocks drive all this physiology? So a major way that they do it is by driving rhythms of gene expression. Now, I told you that those two clock proteins can go and affect their own transcription to generate a clock. But when they go to the nucleus to affect their own transcription, they can actually also affect the transcription of other genes. And so many genes are expressed cyclically under control of the clock. Here is a heat map where the yellow indicates high expression and the blue indicates low expression for several different genes, never mind what they are. And this is over a day-night cycle. So the genes shown up here are peaking in the, towards the end of the day, beginning of the night. The genes here are peaking in the early day. And then there are a few that are peaking in the late night. Now, if you do the same experiment where you look at cycling gene expression in an animal that has a mutation in a clock gene, you see that the expression is random. You lose the rhythmic pattern. So it turns out that when you look at different tissues, like if you look at the heart or the kidney or the muscle, the genes that are cycling in each tissue are actually quite different. The only genes that are in common across tissues are the ones that are part of the clock, and the other genes are different. So the liver, for instance, may be driving rhythms in cholesterol and glucose metabolism, but the pancreas is driving rhythms in insulin expression. And so the specific genes are different in different tissues. So if you look across the whole genome and ask, is this gene cycling in any tissue? There's actually a very high likelihood that it is cycling somewhere. I mean, I've heard numbers as high as 50% that, every gene, that half the genes out there are cycling in at least some tissue. In a given tissue, the percentage of genes that are expressed rhythmically is anywhere from, 5 to 20%. So what I've told you so far is that body clocks are synchronized to light by the clock in the brain. What I didn't tell you yet is that metabolic clocks, not surprisingly, like for instance, the clock in the liver, is more sensitive to the time of feeding than it is to light. And this has huge implications for our fitness and well-being, because what happens then is that if you are eating at the wrong time of day, your liver clock is not going to be in synchrony with your brain clock. And so you're creating this internal scenario of jet lag. Because what happens is that the clock in the brain is most sensitive to light-dark cycles. And so it's going with that. And so it's really important that meal times be in synchrony with that light-dark cycle. Now, many of you have probably heard that, it's really good to do intermittent fasting, or to do what's called time-restricted feeding, where you restrict your food intake for some hours a day. And that actually is a really good thing because I think metabolic processes need to be segregated, and that's part of what the circadian clock does, is to segregate metabolic processes. And one of the ways you can help that along is by restricting your food intake to a fraction of the 24-hour day. But it's really important to time that fraction when you have your food intake so that it's in synchrony with the light dark cycle. So you're not helping yourself by eating for 8 hours, if that 8 hours is 8 p.m. to 4 a.m. as opposed to during daylight hours, because then you are desynchronizing your brain clock and your liver clock, and you're creating internal jet lag. And I've already told you that that internal jet lag is bad for physiology. So synchrony between these different entraining stimuli is really important. And we think that, it's easy enough to synchronize them by just regulating your own habits somewhat. So one of the things that's actually in the field happening now is, a lot of the emphasis in the circadian field for a long time was on basic mechanisms. And now recognizing that there are all these clinical implications, people are looking at how it is that circadian disruption leads to disease. So, it's easy to say, well, this happens, but how does it actually do it? And so I thought I'd just mention some of the translational work that we have done in our lab. And so we found, for instance, that circadian disruption increases tumor growth. And we've actually found the mechanism by which it does that. It's actually through circadian regulation of the cell cycle. In some work we did in collaboration with Richard Stone, we found that circadian disruption increases incidence of myopia. And this, it turns out, is because the circadian proteins are regulating the refractive development of the eye. And it's work that we did actually in flies and in mammals. But we're finding too that circadian regulation can be exploited for treatment. So, not only is it clinically relevant because its disruption can lead to disease, but you can use knowledge of the internal rhythms to treat disease. So one of the things we found in recent years is that the blood-brain barrier is under circadian control. And so drugs that are delivered to the central nervous system, or specific classes of drugs delivered to the central nervous system, are going to access the brain or the central nervous system better at certain times of day. What that means then is that if they are taken at that time of day, you could probably get away with a lower dose and have fewer side effects. And then I told you that circadian disruption increases tumor growth because it's regulating the cell cycle. Well, so its regulation of the cell cycle also then lends itself to timed chemotherapy. Because the circadian clock is regulating the cell cycle, and different aspects of the cell cycle are taking place at different times of day, you can target anti-cancer drugs at the right time of day so that they are actually hitting the process they're targeted to in a more appropriate manner. And we've actually done a screen where we looked at about 150 anti-cancer drugs, and we find that about half of them are working better at a specific time of day. And one of the projects that we have underway right now, actually, which I think your group was contacted about even at one point, because we were looking for volunteers, and that is to look for blood-borne signals that synchronize circadian rhythms, and how those might change with age. So I told you that the clock in the brain is synchronizing the peripheral clocks to the cyclic environment, to the day-night cycle, through several mechanisms, and one of them is through secreted signals. And so we've actually developed a cell culture model for studying the serum from younger and older people. And we find that there are differences in their effects in our cell culture model on rhythms. And we're trying to figure out what these signals are that cause this difference. Because we know, of course, that circadian rhythms change with age, or sleep-wake cycles in particular change with age. And that's been reported in humans, shown here on the right, and also by us in flies. So here in humans, if you look at a young subject, you have a very well-consolidated activity, and then nighttime sleep here, parts in the middle. The older subject doesn't have such well-consolidated activity. There are, daytime naps, and then there are nighttime awakenings, shown by the blips here. And we've done this in flies, and we find kind of the same thing. With flies, because their life cycle isn't so long, we can actually look at the same fly, young and old. So here is a female Drosophila assayed, young, and you see that she's very active during the day and completely inactive at night. And then here is the same fly when she's old, where the pattern has broken down. So one of the questions that we're asking, of course, is, would improving sleep cycles alleviate pathologies associated with aging? So in general, we think that maintaining robust circadian cycles is a simple, straightforward way to improve fitness, wellness. There are many challenges to that, though, in society. And these are some of the other issues. So I told you that, circadian biology was more focused on basic research for a while, and now it's getting more translational. And they are also getting more involved in societal issues. One of them is daylight savings. And I'd be interested to hear what you all have to say about it. I have to say I have mixed feelings about this myself, for many reasons. But from a purely scientific point of view, from the perspective of what happens to your internal clocks, daylight savings is not a good thing. Because we're all being made to shift beyond what our internal rhythm is meant to be. And so many in my field are lobbying, advocating against this. And that's not just in the US, but also in Europe. In fact, I think in Germany, they're making more headway with this than we are here. In the US, it seems like whenever a state wants to get rid of the time change, what they do is try to make it permanent daylight savings instead of permanent standard time. And people in my field are vehemently opposed to that. Like I said, I have mixed feelings about it because I feel like there might be some aspects of daylight savings that are beneficial, even though it's not good for your clocks. But from a purely clock perspective, this is not a good thing. Okay, now we know that circadian, I think everybody talks about this and is sort of vaguely aware of it, that circadian phase does vary from person to person, right? So there are some people who are what we in the circadian field call larks. They are the early birds, they're most functional in the morning, they wake up early and they're active and alert, whereas others are not. And then there are the people, and these people then are probably the night owls. So they're more functional at night. So these are, we believe, genetically determined. Okay, I mentioned advanced sleep phase syndrome before. And so that's an extreme example of what we would call a morning lark, because those are people who are advanced by like several hours. So that kind of, falls into the category of disorders. And they're inherited in a Mendelian fashion. You can study them in families and you can find out the genetic basis. But even in a normal population, there is a wide spectrum. And there are people who are going to be at one end or the other end of it, and the night owls versus the morning larks. And so the thinking is that those disorders like advanced sleep phase syndrome are caused by mutations in clock genes, and that these phenotypes in the quote unquote, normal population are due to polymorphisms in clock genes. So there are naturally occurring polymorphisms that make you early and that make you late. And the reason I'm calling this a societal challenge is because the circadian field would argue that people's jobs should be suited to their internal rhythms. Of course, that's usually not possible. Maybe something like COVID has made that possible, where people are working from home and they can choose their own hours. But when we go back to normal life, that won't be the case. But the circadian biologist would make the case that it should be. And then here's another example of a challenge in society that has many circadian people actively politically involved, and that is the timing of schools. So as you all are aware, the way schools are timed here is the start times are staggered for elementary schoolers versus middle schoolers versus high schoolers, with the high schoolers going to, and they have to be staggered because of the bus schedule. But they have the high schoolers going to school the earliest and the elementary school kids going last. And that actually is the exact opposite of what should be, because adolescents are notoriously delayed in their circadian phase. So they have a circadian phase delay, they have a slow rise of sleep pressure. In addition, they may have other things that keep them up at night, academic pressure, screen time, social networking. But the bottom line is that everything in them is pushing them to be like a night owl. And yet, society has them waking up really early to go to sleep. And that is not just disrupting their rhythms, it also stops them from getting enough sleep. And as I'm going to tell you in just a minute, sleep is really important, especially in younger life. So there have been efforts, even in our area. Lower Merion actually came close to changing school times. I even got contacted by some of the parents there who were concerned about this. And at the end, I think they have backed away from it because they are getting a lot of pushback from some parents, which the people who are advocating for this are not happy about. And some of those are actually our colleagues at Penn who've been pushing to get these school times changed. Okay, so what I'm going to do in the next part is actually switch from talking about circadian timing to talking more about sleep. So if there are any pressing questions, I'd be happy to take them. Oops, I think I saw something in the chat. Do you alter your fast schedule based on change of daytime light over the different seasons? The fast schedule, I assume you mean someone who's naturally advanced? The short answer is that yes, the schedules do tend to change over the seasons. We don't exactly know why that happens. So, seasonal timers are different from circadian timers, but schedules do tend to change. And of course, in the very northern climates where you have 6 months of dark or 6 months of light, circadian rhythms are quite disrupted. Another question, is there a way to understand your own rhythms other than feeling like a day or night person? There is, yes. So you can actually get yourself tested for melatonin. That is the best marker in saliva. And you would have to do that at different times of day to see what your internal rhythm is like. Matthias Spasner, who's one of my sleep colleagues, says Lower Merion is now contemplating to start with middle school, but the whole initiative got drowned out by COVID-19. Yep. Yeah, COVID's of course complicated everything. Thank you, Matthias. Is there a particular time during the day that the blood-brain barrier is more amenable to passing from circulation to CNS? Yes. So I didn't get into the details of this, but the process we've looked at that is subject to circadian control is the passage of small lipophilic molecules. So these are molecules that can easily diffuse through the lipid bilayer of, the cells of the blood-brain barrier and can get into the brain. And it turns out that there are efflux transporters that actively pump these molecules out of the brain. And those efflux transporters are more active during the person's waking hours. So the diffusion into the brain is better than during the sleep hours. And many drugs are these small lipophilic molecules. And we showed, for instance, that phenytoin, Dilantin, the drug used to treat epilepsy, is one of these molecules that gets into the brain better during sleep. Do I recommend taking melatonin? Actually, I am not a big fan of melatonin. I think there's a lot of hype about it. It's definitely, it resets your clock by a little bit, not nearly as much as people claim it does. For those who take it, they'll say, oh, I took it and it was totally fine. And some of that I think is placebo. It does help to put you to sleep in the short term, okay? It's not necessarily going to keep you asleep all night, but it will put you to sleep for a few hours. And you have to be very careful about the dose you take. So I think a lot of people take 3 milligrams, and I'm forgetting about the exact numbers, but I think that the amount you should take for the hypnotic effects is actually a little bit less. Do I advocate for the use of bright lights to simulate daylight for those who cannot get morning sunlight? Yes, definitely. But nothing compares to sunlight. So if you can get sunlight, you by all means should get sunlight. In fact, the best way to treat jet lag is to get out into the sun, to adjust to the new schedule. Okay, I'll keep going, I guess, and we can also do questions after I finish with the next one. Part of my talk, which has to do with sleep. So when it comes to sleep, so the interesting thing is, you might think sleep is a circadian rhythm, but actually these are two separate fields with two separate journals and separate conferences. Circadian rhythm and sleep's very different. In my lab now, we do study both. And when we say we study sleep, then we're not talking about the timing of sleep, okay? Because it's not just the timing of sleep that's important. So even if our sleep was not timed, we would still need to sleep. And you see that, of course, if you've been up late at night or been up all night, and in the morning, you want to sleep in, although your internal clock and your alarm clock are telling you to wake up. And that is because we all have this need to sleep. And that need to sleep can sometimes overcome the circadian clock. And so this aspect of sleep, which we refer to as the homeostatic control of sleep, is the system that ensures that an organism gets enough sleep. So just like there's a homeostatic need to eat, if you haven't eaten in a while, you need to eat. There's a homeostatic need to sleep. So if you haven't slept in a while, you need sleep. And so when we study this homeostatic control, what we're looking at is the duration of sleep, not the timing, the duration of sleep, the depth of sleep, and how we respond to sleep deprivation, like how quickly we fall asleep when we haven't slept, like how strong is our need to sleep. Now, what makes us sleep and why do we need to sleep? And the short answer is we don't really know. We spend a third of our lives sleeping, or we should spend a third of our lives sleeping. I know many of us don't. Actually, I do. But we still don't know what's so important about sleep. We do know that sleep need varies. So it varies across age, from newborns who need a lot, infants, toddlers, whatever, preschoolers, all the way to adults who need less. And even in adults, it varies a lot. So there are some people who actually do need less sleep. There are others who think they need less, but they're usually deceiving themselves because they're crashing over the weekend. But the interesting thing, and this is one of the things that we've actually addressed in our fly model. So I didn't get into the details here, but when I first started my lab, we were using Drosophila to understand circadian rhythms. And we were looking at their activity and rest as readouts of their circadian clock. And then at some point, we became interested in whether that rest in flies was sleep. And we basically established it as a model for sleep. And so we've been trying to do a lot of different things with it, just to touch upon some of them. So all young animals sleep a lot. And so do young flies. And so one of the questions we addressed was why young flies need to sleep a lot. And so this was work done in my lab that Matt Kaiser, he was a postdoc who now has his own lab in the psychiatry department at Penn. And what he did was to take really young flies that sleep a lot, and he deprived them of sleep. And then when they were adults, he asked if they were impaired in any way. And he found that they were impaired in their ability to court. So flies have this courtship ritual, where the male courts the female, and he goes through these very stereotyped steps where he orients towards the female, he taps, he sings a little song by flapping his wing, and then he licks, and then there's copulation. And so what Matt found was that when he deprived flies of sleep when they were really young, they were impaired in their ability to court later in life. And not only were they impaired in their ability to court, but he found that even those that courted were not actually making it all the way to copulation. So copulation frequency was greatly reduced. And I'm going to play a little video here where you can actually see this quite visually, where the flies on the left are control flies. So they slept when they were young. And the ones on the right show the male chasing the female, and he was deprived when he was young. And you'll see here that the flies on the left, do pretty well. So this male now is already mounting and copulating. And the male that was sleep deprived when young sort of gets distracted. He pursues the female. There's nothing wrong with him in terms of locomotor ability. He does this, but it's very half-hearted, and he never quite makes it to copulation. Okay, I will stop that and move on. So Matt found that when he deprived flies young in life, that they did not court very well when they were older. And he found that it's because the circuits that drive this behavior don't develop as well. So we believe that sleep is required in early life for the development of circuits that drive adult behavior. And in this case, it's a behavior that's very important. It's required for species propagation. So why is sleep important otherwise? Why is it important in adults? And one of our approaches to that has been to isolate sleep mutants. And shown here is the first mutant we got out of a sleep screen isolated by my postdoc, Hyung-Hee Ko, and her colleagues, Bill Joyner and Mark Wu. All these people now have their own labs, actually. So here we're looking at sleep in male flies and in female flies over a day-night cycle. So we look at activity, and sleep in a population of flies. We calculate the amount of sleep, and we plot it here as an average. So male flies have a huge afternoon siesta, and then they have nighttime sleep. Female flies have less of a siesta, and then they have very well consolidated nighttime sleep. So you can actually tell from looking at a record whether it's male or female, because males have a much more pronounced siesta. And this mutant we got, though, sleepless or SSS, it has reduced sleep. It reduces sleep in males and females dramatically by like 80%. And so why are we isolating these mutants? We're isolating these mutants because we're thinking, well, they could tell us, what drives sleep. They might even tell us why we sleep. So for instance, if you get a mutant that sleeps less and has some pathway upregulated, then maybe that is why they need to sleep less, because that pathway normally occurs during sleep. But if you already have high levels of it, you don't need to sleep that much. And I'll just tell you that, the short sleeping mutants that we and others have isolated so far through unbiased approaches in Drosophila, because that's what Drosophila really lends itself to, is unbiased approaches. They all, for the most part, are neuromodulators. Okay, so they are affecting the signaling of neurotransmitters, they're affecting membrane excitability. And to make a very long story short, right now, the way we are thinking about this is that the reason these mutants are not able to sleep is not because they don't have the will to sleep or the need to sleep. So they are not the kind of short sleeper you want, where they have less need to sleep. We believe they actually have the need to sleep. They are not able to sleep because they are not able to modulate their nervous system to fall asleep. So they are genetic models for insomnia. Okay, so how then do we get at, so nothing here is telling us why we need to sleep. We think that these are just molecules that are sort of downstream of the need to sleep, and they modulate the nervous system. So nothing here is telling us why we need to sleep. So how else do you get at this question? Well, you can sleep deprive the animal or the person and ask what goes wrong. And I've already told you, that circadian disruption leads to metabolic effects, so does sleep loss. But then there's also impaired immunity, cognitive impairment, decreased reaction time. A lot of these studies on cognitive impairment and decreased reaction time have actually been done by my colleague, David Dinges at Penn. He's like, the leading lab on these types of studies. So we teamed up with him because he does these studies with humans. He brings people into his lab, he deprives them of sleep, and then he looks at these functions. So we teamed up with David Dinges to look at molecular markers in the blood. And what we found was that, so in human subjects, many metabolites in the blood are altered by sleep loss. And in general, what we find is an oxidizing environment and signs of insulin resistance. Now, you may have heard this, that sleep loss, makes you, increases your susceptibility to diabetes. That is supported by these data. So what we're finding is that loss of sleep has a huge effect on metabolism. And what I'm going to do in the last couple of minutes is basically tell you that we now have data from several different projects in the lab that support the idea that sleep is required for metabolic homeostasis. Now, when I'm talking about metabolic homeostasis here, this is not just about, diabetes and insulin or glucose homeostasis. I'm talking about cellular metabolism, cellular energetics. So the brain, consumes a lot of energy, and then that energy has to be replenished, and you need metabolic energetic pathways. And that's the type of metabolism that we think sleep is important for. And here are some of our recent data that support the idea. So I told you about the blood-brain barrier being more permeable to peripheral molecules at a specific time of day. And that is under circadian control. Okay, so that is controlled by a clock. What we found through separate studies is that endocytosis with the blood-brain barrier, which is taking stuff up from the brain, is dependent on sleep. Not on circadian timing, but on sleep. And we believe this is to clear metabolic waste. Now, I also told you that we've been looking for short sleeping mutants because we think this might help us get at the function of sleep. But so far, we think those mutants are they're not sleeping because they can't modulate their nervous system, but they have the need to sleep. But in very recent work, which is not published yet, we actually found a mutant that affects autophagy. And autophagy is the process by which clearance of metabolic waste takes place within a cell. And so now we're finding that sleep is important, not just for clearance of metabolic waste through the brain on an organ level, but also on a cellular level. And finally, in work that is actually going to be published tomorrow, although it's already online, we find that flies need sleep to consolidate appetitive memory if they are fed after training, but not if they are starved. So appetitive memory is basically, the paradigm is you teach the fly that sugar is associated with a particular odor. And then they remember and they always choose that odor. And that type of memory about where food is located, flies are able to consolidate that even when they don't sleep. Okay, and that makes sense because when animals are starved, they actually have to go foraging, they have to go looking for food. So they don't have the luxury of sleeping. And under those conditions, they are able to remember cues about food without sleep. On the other hand, if they are fed, then they need to sleep. And we think that that's because feeding them is triggering metabolic reactions that then require sleep. Okay, so basically, my last couple of slides is just, takeaways from this talk. I think that circadian rhythms, improving the circadian rhythms is a very easy, non-invasive way to improve health. And the way you can do this is with, somebody already asked me this question, robust cycling with bright light during the day, dark at night, get enough sleep, improve metabolic rhythms by restricting timing of food intake. And here is where we are headed as a field. So a couple of hospitals in the country have already done this. In fact, CHOP is one of those hospitals where in 2 units, they have lights that can be timed and that also have different wavelength to simulate dawn, dusk, and midday, because they are recognizing that those cues are important for wellness. And so some of us have been talking to Kevin Mahoney about having such a system in the new hospital at Penn in the Pavilion. Now, eventually, of course, this by itself won't work. You also need to have appropriately timed physician rounds. You can't have the doctor waking you up at 5 a.m. to take some measurements. We are advocating for drug delivery timed to the patient's internal rhythms, in part because we know that a lot of drug targets cycle, in part because we now know about rhythms in the blood-brain barrier, for instance. And given that circadian rhythms can be different from person to person, treatment tailored to each individual would be way more beneficial. And that is what precision medicine is about. And then finally, we would say that even outside of healthcare, having jobs, duties assigned based on internal rhythms would be a productive way to go. And I will stop there and be happy to take more questions. Okay, so what are some of the daily practices you have in place to optimize synchronization with your circadian rhythm? So the daily practices are get a lot of sunlight as much as you can during the day. Try to maintain a regular schedule in terms of when you wake up and when you go to sleep, and have it be as much in sync with the day-night cycle as possible. And then restrict your timing of food intake. So stop eating 3 hours before you go to bed. And don't try to restrict your feeding. I mean, ideally people say 10 hours. I think even 12 hours is okay, but try not to go beyond that. Okay, please expound on the statement that sleep is different from circadian rhythms. Yeah, so circadian, this is a question that always confuses people. So circadian rhythms are basically cycles of 24 hours. And they occur, like I said, in almost all of physiology. Heart rate, blood pressure, body temperature. Sleep is just one of those processes that has a circadian rhythm. And when we say we study sleep for itself, what we are interested then in is not in the timing of sleep, but in the amount of sleep, the depth of sleep, how much sleep do you need, how do you deal with sleep loss? And that, I hope that answers that question. What about midday napping? Midday napping it's actually fine. I actually, believe that humans are meant to take siestas. And there is a reason that the coffee carts have so much business in the afternoon, especially the postprandial dip, right after lunch. And, sometimes your job doesn't allow you to do that, but all said and done, it's better to take a nap than it's better to have well-consolidated sleep at night. Let's just put it that way. But if you're not able to get enough sleep at night, it's better to take a nap than to not take a nap. Can I comment on sleep debt and gut dysbiosis? Yeah, so this is actually one of the studies that we also did in collaboration with David Dinges, where we took people who had been allowed to sleep for only 4 hours a night for 5 nights. And then we looked at their gut microbiome. And we found that it was actually fine. It was not affected by 5 days. So, we were not doing total sleep deprivation because we wanted to mimic a real world scenario. Most people are not sleeping at all. They're just sleeping less at night. And so that was why we followed the protocol we did. And that, 4 hours of sleep a night for 5 nights did not affect their gut microbiome. Since then, people in Colorado, our colleagues in the field, have taken this out further, and they basically reproduced our findings that that protocol does nothing. But if you go longer, then you have gut dysbiosis. Oh, and can I comment on pancreatic sensitivity? I don't remember what the phase is, but insulin cycles. I'm guessing that it is in conjunction with, glucose intake. I would have to look it up. And so it would be higher, more insulin produced during the day for us, but I don't remember offhand, but it is rhythmic. Are age-related changes in sleep cycles inconsistent with good health? Yeah, that's a good question. I have a circadian colleague who keeps asking, why are we studying aging? It's a natural process. Everything that happens with age is fine. And I don't know that that's true. I think that there are some people who have better sleep cycles, who maintain good sleep cycles with age. And I think that that does associate with better health. I should say too that in this project we just did, that we're currently still doing, it's work in progress, where we took serum from older and younger people and looked at the ability to reset rhythms. We found that the old serum actually does quite well. But I think it's also because we were selecting subjects who were older, because we were, who were healthier, because we were taking people who were not on any drugs, any medication. So we were naturally selecting for healthier people, and then they were also better at synchronizing rhythms. We're at 1 o'clock already, but there are more questions. Do you want me to keep going, or?
Yeah, I think it's fine. For people
Okay.
who want to leave, they can leave, but I think people are fascinated by what you're telling us. So if it's okay with you.
Sure, yeah.
Can you?
Okay, how to stay in sleep, especially during aging. So keep your rhythms as robust as possible. Again, try to get lots of bright light during the day. Try to maintain a regular bedtime, control the eating so that you stop 3 hours before you go to bed. You can try to, try melatonin. It's harmless, and it works for some people. That would be another way to do it. How does drinking alcohol in the later evening affect sleep? So what alcohol does is, of course, the effects of alcohol vary from person to person. So, some people get high with it and some people get drowsy with it. But even when it works a bit of like a sedative, it's only for the short term. So you'll fall asleep, but then you'll be up a few hours later. So it won't have this effect of keeping you asleep all night. The effect of caffeine and alcohol. So caffeine is a big disruptor of sleep, and not just in humans, but even in our fly models. It's actually one of the ways we can use to deprive our animals of sleep. Are you asking caffeine and alcohol together? I already answered alcohol. I don't know about caffeine and alcohol together. One of the independent, yeah. So the alcohol one I already said, it may put you to sleep in the short term. Caffeine will wake you up. It's a very, but you will crash when you come off of the caffeine. So that's one of the downsides of caffeine, is it'll keep you up as long as it's in your system, and the minute it wears off, you will crash. So the search is for a drug where you will not have a rebound. Read about decreased stage 3, 4 sleep. Yeah, these days people tend to talk in terms of 3 stages of sleep rather than 4. So slow wave sleep is affected by caffeine, which is what that would be.
Well, it looks to me like you've addressed all the questions, and I want to thank you so much, Dr. Sehgal, for a fascinating talk, and also for giving us great advice about how to try to sleep better. Thank you so much. This has been terrific.
Thanks for having me. All right, that was fun.
You.
Bye.
Bye.